Aloha everyone! We are finally going to get out of basic chemistry and basic biology today, and we're actually going to get into anatomy. So we are going to begin our discussion of chapter 4 in your e-text, which is all about our tissues. So in chapter 3 we talked about cells, and when a group of cells that are similar in their structure come together to perform a common function, we call that a tissue. So that's what we're going to be talking about in this lecture as well as the next, is all about our tissues. So today, I'm going to start off by looking at our key questions and learning outcomes. And then it says here an epithelial tissue mini lecture. So what that means is today's lecture is going to be a little bit shorter than normal. It's not going to take up that whole 1 hour and 20-minute class period. And the reason for that is at the end of the lecture, I kind of go over some of the information for one of your assignments this week, which is your epithelial tissue POGL assignment. So I kind of walk through some information that will help you answer some questions on that, and then give you some extra time today to work on that activity. Because if we actually were in class, that's what we would be doing. I'd do this short lecture, and then you'd have time to work on that activity. Starting off, let's look at these key questions. First of all, what are the different types of cell junctions? What are the functions of these cell junctions? What are the 4 different types of tissue? And what are the specialized characteristics of our epithelial tissue? So by the end of today's lecture, you should be able to answer these key questions. Here are your learning outcomes for this lecture. You can see there's a lot of them, right? Even though it's a short lecture. So we're going to start by talking about cell junctions, defining the term histology, listing the 4 major tissue types. We're going to compare and contrast general features of them. We're going to define a lot of terms talking about cells. We're going to talk about the different cells that come together to make our epithelial tissue and the distinguishing structural characteristics of epithelial tissue. And that's kind of what that POGL activity that you'll have time to do at the end of lecture is really mostly about, is this epithelial tissue and the different types of epithelial tissue. So let's go ahead and get started. So I'm going to start this lecture off by talking about cell junctions. And if you've read through chapter 4, you might be like, wait a minute. I did not see anything about cell junctions in chapter 4, and you would be correct. So cell junctions were actually talked about in your e-text back in chapter 2, and I didn't cover them in chapter 2 because I felt like it was a little out of place there, right? Chapter 2 was all about chemistry. So now when we talk about tissues, we're talking about a whole bunch of different cells that perform the same function or at least a similar function. And the cells are going to group together to create tissues. And then the way they group together is by using these cell junctions. So when we talk about tissues, which is the subject of chapter 4, right, it makes more sense to talk about the connections between cells that actually allow them to form tissues. So that's why we're going to be talking about these cell junctions here. So humans are very complex organisms, right? We're made up of trillions of cells, each with its own structure and function, and cells that do the same job, again, are going to combine together to form body tissue, such as our muscle tissue, our skin tissue, our bone tissue. Groups of different types of cells are going to make up the organs in the body, such as your heart, your liver, or lungs, and then each organ has its own job to do, right? But then all those organs are going to work together to maintain your body. So most cells within the tissue are going to stay in place to create that tissue because they're anchored to other cells through these cell junctions. They're anchored to a basement membrane, which we'll talk about in a little bit, and it's kind of like another layer. It's like an anchor, but it's not just another cell, because really it's a membrane and it is continuous. So that basement membrane is not a cell, it's kind of a membrane, just like the term says, basement membrane. It's going to be continuous. Other cells are also going to remain in place because of connective tissue, which we'll talk more about in our second lecture, lecture 4B. Each of the cells have their own structure and function, right? So also there are going to be cells that are actually free. So they're not going to be bound to any other cells, such as think of our blood cells. They're going to float freely in our blood. Or sperm cells, those are going to float freely in the seminal fluid. So they're not joined to anything in particular, but most cells are going to be bound together again to form tissues and organs. So again, cells are that basic unit of life that we talked about in chapter 3. They're going to bind together to create tissues, and different tissues put together will create an organ. Different organs come together to create an organ system, and then those organ systems all work together to maintain our life. So next we're going to talk about these 3 ways listed here that cells can be bound to one another. And these 3 categories are tight junctions, desmosomes, and gap junctions. So on the following slides, we're going to go through each one of these in more detail. The first type of cell junction that I want to talk about are called tight junctions. And these tight junctions are, well, just like what they sound, right? They're tightly put together. So we have integral proteins on adjacent cells, so adjacent just meaning they're right next to each other, right? They're going to fuse to form an impermeable, meaning that things cannot pass between them. So it's an impermeable junction, nothing's going to get through there. And that's going to encircle that whole cell, which prevents fluids and most molecules from moving in between the cells. So tight junctions are primarily going to be found in sheets of epithelial tissue, which cover the surface of our bodies, right? And they line internal cavities. So all epithelial sheets are going to be highly selective barriers between 2 compartments which with considerably different chemical compositions. Again, tight junctions are going to be impermeable junctions that join together the lateral edges of your epithelial cells near their borders, and this prevents movement of materials between the cells. So only regulated passage of materials can occur through these cells, which form highly selective barriers that are going to separate the 2 compartments of highly different chemical composition. So again, when you think of these tight junctions, think of our skin, right? These are found in our skin, and we're going all over the place each day usually, right? We don't want bacteria and viruses to be able to get underneath of our skin and compromise the rest of our body. We also find these tight junctions here in our stomach and intestines because we have digestion and everything that's going on in our intestines, and we really want tight contacts in the small and large intestine because we don't want the gastric juices or the other fluid to be able to pass freely in and out of the gut. You may have heard of something like leaky gut, where we're having some of those contents leak out and damage other components. We don't want that very acidic gastric juices to be going into other organs. So we want to make sure that there's, again, kind of a bouncer there, right? Only allowing the nutrients in and not the harmful bacteria and other stuff in. We don't want other fluids to leak into our intestines, right? Because then that'll get transferred to the rest of our body, and say it's a bacteria or a virus, that's how things are going to spread really quickly. So again, here you see what these tight junctions look like. So you can see these cells, these are actually epithelial cells right next to one another. And this is kind of blown up to show you this interlocking junctional protein. So this is a tight junction here. And keep in mind, something I want to point out is you're going to see this figure on the next 3 slides, because this actually has an example of all 3 types of cell junctions. So keep that in mind. The one shown here is right here. this tight junction. So it's between these adjacent cells, again, right next to one another, and you can see it's very tight. Impermeable junctions. It forms this continuous seal around the cell. It's going to prevent those molecules from passing through. Again, commonly found in our skin, also our stomach and intestines. The second type of a cell junction is a desmosome. I know, fun name, right? So a desmosome is a rivet-like cell junction, and it's formed when we have these linker proteins that are known as cadherins. So those cadherins of the neighboring cells are going to interlock kind of like the teeth of a zipper. So the linker protein is going to be anchored to the cell through this thickened button-like area on the inside of the plasma membrane, and that's called a plaque. So the keratin filaments are going to connect those plaques intercellularly for added anchoring strength. What this does by having kind of that zipper is it allows a little bit of give between the cells. So that reduces the possibility of tearing under tension. Right? So if you look here, this is blowing up one of these desmosomes. You can kind of see how it looks like a zipper, right? Right here. And again, in bright blue, these are our linker proteins, our cadherins that kind of form that zipper-like structure. Then on the side, you have this button-like structure. That's going to be your plaque again, right? So that's keratin filaments are in purple here. Those keratin filaments, again, are going to connect these plaques intercellularly for added anchoring strength. So that's what your desmosome looks like there. So where might a desmosome be useful in the body? Well, these intercellular filaments are going to bind adjacent plasma membranes together. And the purpose of that is that's going to allow them to resist being pulled apart. These desmosomes are going to be our strongest cell-to-cell connections. So desmosomes are going to be most abundant in tissues that are really subject to considerable stretching, such as those found in our skin, in our heart. Think of just when your heart is contracting, there's a lot of stretching going on there, right? When it contracts, and each time that your heart is beating. Also, if you think of the uterus, every time that a child is born, right, there's a lot of stretching of that uterus as well. Additionally, if you think of our cytoskeletal filaments, such as the tough keratin filaments in our skin, they're going to stretch across the interior of the cells and attach to that desmosome plaque located on the opposite side of the cell's interior. So again, this arrangement of these desmosomes forms kind of this continuous network of very strong fibers throughout the tissue. So again, we're going to find these in places that are subject to a lot of stretching. And you can see, I think over here, it's showing you a cardiac muscle cell here. So one of the places we see a lot of stretching is the heart, uterus, and our skin. The final type of cell junction I want to talk about are these gap junctions. We have these transmembrane proteins known as connexons that form almost these tunnels that are going to allow small molecules to pass from cell to cell. So these gap junctions are used to spread ions, simple sugars, and other small molecules between the cells. So this allows electrical signals to be passed very quickly from one cell to the next cell. And it's used a lot in our cardiac muscle cells as well as smooth muscle cells. So gap junctions really are communicating junctions. The small diameter of the tunnels is going to permit, again, your small water-soluble particles to pass between the connected cells, but it precludes or it stops that passage of your large molecules. So ions and small molecules can be directly exchanged between interacting cells through these gap junctions without ever having to enter the extracellular fluid. Gap junctions are especially abundant in our cardiac muscle and our smooth muscle. So in these tissues, movement of the ions through gap junctions is going to transmit electrical activities through an entire muscle mass. So because this electrical activity brings about that contraction, the presence of the gap junctions is going to enable a synchronized contraction of a whole muscle mass, such as the pumping of a chamber of the heart. So we're going to see the importance of these gap junctions throughout this course. So here you're seeing the bottom right here is an example of one of these gap junctions. You can see the space between the cell here. You see these connexons, so those are those transmembrane proteins, right? So you can see how there's that little space. It's going to allow those ions and small molecules to be directly exchanged there. Okay, now we're going to move on from talking about those cell junctions that were covered in your e-text back in chapter 2, but I think fit in more with what we're talking about today. So those cell junctions are used to connect cells, right? And cells that have a similar structure, they're going to come together to perform a similar function. And that is what our tissues are. And tissues are the subject of chapter 4 in your e-text. So we're going to begin chapter 4 today and then finish it in lecture 4B. So as mentioned before, those cells of similar structure and function will connect together to form a tissue. And that's going to be what chapter 4 is all about. So tissue is, as it says, kind of our living fabric. Each of the individual body cells are going to have their own specialized function in order to maintain homeostasis. But for tissues, there are groups of cells, again, that are similar in structure, that perform a common or a related function. Histology is going to be the study of tissues. So histo actually means tissue or anything that stands up, and anytime you see ology, that means a branch of science, right? So histology is going to be the study of tissues. We have 4 basic tissue types, and you can see them here. We have epithelial, connective, muscle, and nervous tissue. So if you had to put into one word what each of these types of tissues does, I would say that your epithelial tissue is going to be covering. Your connective tissue is mostly used for support. That muscle tissue allows for movement, and our nervous tissue is going to control. But you're going to see very quickly that we really can't just use one verb to describe each of these tissue types, right? Each tissue does a lot more than can be put into a one-word definition. So this figure here is from your e-text, and it actually outlines the 4 basic types of tissue. So first on the top here, you see your nervous tissue. Nervous tissue is used for internal communication, and it's going to be located in areas such as our brain, our spinal cord, and our nerves. Nerves are usually shown in yellow on figures. So if you see kind of these lines that are bright yellow, that's usually how the textbook shows you nerves. We'll talk a lot more about nervous tissue at the end of the semester. We'll also talk about it more in lecture 4B. So secondly, we have our muscle tissue, which is contractile tissue. We have muscles that are attached to the bones that are going to move our body. They're used for locomotion. We also have muscles in our heart, right? That's going to be our cardiac muscles. They're different. They look different than our skeletal muscles under the microscope. Then we have the muscles of our hollow organs. So that's going to be things like our small intestine and large intestine and stomach. And those are known as our smooth muscles. And what they do is they help with digestion to kind of push along all of the nutrients in your digestive tract. So we'll talk more about muscle tissue later as well. Then we have our connective tissue. So connective tissue, you see down here. That connective tissue helps to support, protect, and also bind tissues together. So this includes things like our bones, tendons, fat, and other soft tissue padding. Fourth of all, we have epithelial tissue, and this is actually the only tissue type that we're going to talk about in depth today. We'll talk about the rest in lecture 4B. So the epithelial tissue, when you think about epithelium, I always kind of think about skin, right? But epithelial tissue is not just our skin. Epithelial tissue is going to form boundaries between different environments. So it's going to protect, secrete, absorb, and filter. So places in our body that we find epithelial tissue include things like the lining of our digestive tract organs, other hollow organs, our glands, as well as our skin. So again, today we're going to focus on epithelial tissue, and we're going to cover the rest of chapter 4 in lecture 4B. So looking at epithelial tissue again is a sheet of cells that's going to cover body surfaces or cavities. And we have two main forms of this epithelial tissue. One form is going to cover and line the epithelia, which is basically your skin. It's on the external and internal surfaces. So that's this type 1, the covering and lining epithelia. Then we have our glandular epithelia. And these are going to be, this is going to be our secretory tissues, right? So they're going to secrete in your glands, such as your salivary glands. Again, the main function of our epithelial tissue is for protection. They're also going to absorb. So if you think about in your digestive tract, they're absorbing nutrients. So the lining of your digestive tract is also going to have epithelial tissue. Again, it's not just our skin. Epithelial cells are also going to be used for filtration, excretion, secretion. So that epithelial tissue is going to be lining our kidneys and the tubules of your excretory system. So in this picture here on the left in pink, we have a picture of the epithelial tissues in your skin. So you can see kind of how we'll talk more about the different types of this epithelial tissue at the end of lecture. But you can see how these cells are close together, right? That's going to be in our skin. And you can see, even though over here on the right, this is still epithelial tissue, this is actually a picture of epithelial with ducts, right? So you can see these ducts here. The ducts are going to be circular, and there's going to be these little hollow white portion in between them. So again, this is histology, right? This is the study of these tissues. We're looking at these cells underneath the microscope and looking at how different they look, even though these are both epithelial tissue, right? This one is our skin, whereas over here we see a type of glandular epithelia. So still both epithelia, but look very different underneath the microscope, right? So first, we are going to talk about the special characteristics of epithelial tissues. And there are 5 distinguishing characteristics. So they're going to have polarity, they have specialized contacts, they're supported by connective tissues, They're avascular but innervated, and they have a really great regeneration rate. And if some of this looks like, wait a minute, what in the world does this mean? Don't worry, we're going to look at each one of these special characteristics on the following slides in more detail. The first special characteristic of our epithelial tissue is going to be polarity. And we've talked about polarity before. Polarity can mean that there's poles, right? There's opposites, right? But specifically for epithelial cells, it means they have a top and they have a bottom. And we're going to refer to one side, the tops, or one side as being the apical surface. That's going to be the upper or the free side that's actually exposed to the surface or the cavity. Apical can be considered like the top generally, but sometimes images will be flipped, so just be careful about always thinking of it as the top. It's going to be the free side, if that helps you remember that better. Most apical surfaces are going to be smooth, but some will have special finger-like projections called microvilli, specifically in the small intestine. So you can see up here, it's showing you your apical surface. Again, generally thought of as kind of the top. It's going to border the open space. So you can see this open space up here, it's bordering it. So that's your apical surface. Then we have the basal surface. And this is going to be the lower attached side. This is going to be attached to a basement membrane or underlying connective tissue. And this is going to face inwards towards the body when you look at it under a scope. So in this picture, again, you can see the apical surface up here on top. It is exposed to the surface. Then we have this basal surface down here. This is next to this underlying connective tissue layer down here. And it's often going to attach to something we call the basal lamina, which we'll talk more about in a few seconds. But the basal lamina is kind of this adhesive sheet that is going to hold the basal surface of the epithelial cells to the underlying cells. So both our apical surface and our basal surface are going to differ in structure and function. And because we have both this apical and basal surface of our epithelial cells, it's often said that epithelial cells are going to exhibit apical basal polarity, meaning they have those two distinct surfaces. So the second characteristic is with epithelial tissue, we're going to have specialized contacts because the epithelial cells need to fit very closely together. So this second special characteristic of epithelial cells is specialized contacts such as our desmosomes and tight junctions that we already talked about earlier in today's lecture. So hopefully now you understand why I waited to bring those up. So we have the desmosomes and the tight junctions. And if you remember, tight junctions are impermeable, right? And those desmosomes are they have that kind of zipper-like structure with the plaques on each side, and they allow for stretching. So we have the desmosomes and the tight junctions because we want to keep things nice and tight, but we also want to be able to withstand the friction and the pressure that can happen potentially from trauma. So many of these are also going to form continuous sheets. You can see here, again, the picture you saw earlier, they're going to have these tight junctions, they're going to have these desmosomes. They will not have your gap junctions. You're going to see, well, they will have the gap junctions, but they mostly are going to have these tight junctions and your desmosomes. The third special characteristic is that all epithelial sheets are going to rest upon and be supported by connective tissue. So you can see this connective tissue here. It's kind of in this pinkish color down here, right? That's your connective tissue. It's not epithelial tissue. we have up here your epithelial tissue, right? So it's resting on this connective tissue. And we also have this area that is between the epithelial and the connective tissue that really helps to reinforce the epithelial sheet and helps it to resist stretching and tearing. And that area is known, it's right here, it's the other area I highlighted, it's your basement membrane. So we have different components of this basement membrane, so I'm going to kind of break it down a little bit. First we have, you can see it here, it's said to be your reticular fibers. So that's your reticular lamina. So it's actually deep to your basal lamina, and I mentioned the basal lamina a little bit earlier and said I'd talk more about it. That basal lamina, you can see it pointing to it right here, it's this area. So the basal lamina is basically a membrane or a lining, and there are going to also be these supporting reticular fibers that help to make up this reticular lamina. So reticular fibers are going to be a network of collagen fibers, and they're really, really strong to help provide support. So that's this basal lamina here, these reticular fibers. Then the basal lamina and the reticular lamina, you can see they kind of come together to create what we call that basement membrane. And that basement membrane is continuous. It acts kind of like an anchor for the cells to basically stick to it so they don't float away. So the basement membrane serves to reinforce the epithelial sheet. It's going to resist stretching and tearing, and it really defines that epithelial boundary. So if you look at this image again, you can see your epithelial tissue is up here. You have your basal region of it. So then below that, you have your basal lamina, you have your reticular fibers that make up that reticular lamina. The two of them together are making up this basement membrane. Right below that basement membrane, you have your connective tissue. Everything in pink here that is supporting your basement membrane, it's supporting that epithelial cell. So your basement membrane is what's going to define your epithelial boundary, and then you have that connective tissue beneath it. So you may be wondering, why are you spending so much time talking about this basement membrane? Anybody have an idea why we need the basement membrane? So what do you think would happen if that basement membrane fails? What happens if that connective tissue boundary, so where that basement membrane meets the connective tissue, what if that fails in our epithelial tissue? So again, that basement membrane is so important because it is responsible for containing mutant and eventually cancerous cells. So if the basement membrane fails, the mutant cells are going to penetrate into that connective tissue more easily. So they're going to go right through into the connective tissue, and then they're going to more easily spread. So as you can see on this top figure here, the normal cells are going to be this light yellow color, and then the cancerous cells are going to be this orange color. And if you notice in red here, that's your basement membrane, right? You can see that this basement membrane is containing the cells, they're staying in the epithelial. So if your basement membrane integrity is broken, then those mutated DNA cell, or those mutated cells, are going to penetrate into that connective tissue. So you can see that down here. They've gone through your basement membrane. They're now into that connective tissue. And they're going to be able to continue to divide with that mutant DNA and then spread all over your body as shown here. So they're going to be disseminated throughout your body. They're going to penetrate this boundary and then invade underlying tissues, resulting in the spread of cancer. And as a reminder, we talked a little bit before when we talked about the cell cycle, what cancer is, right? And remember, it happens during this S phase where we have DNA replication. So cancer is when DNA replication goes wrong. So DNA replication over trillions of cell divisions can end up with some genetic mutations, right? And those DNA mutations can disrupt the cell cycle. Mutations can be caused by things like radiation, smoking, pollutants, chemicals, and viruses. And there are several different factors that are going to regulate the cell cycle and help to assure that a cell divides correctly. So before a cell divides, the DNA is going to be checked to make sure it has replicated correctly. If the DNA does not copy itself correctly, generally a gene mutation is going to occur. So while normal cells are going to stop dividing if there's a mutation in the DNA, that's not the case with our cancer cells. Cancer cells will actually continue to divide with mutations. And due to DNA mutations, cancer cells are going to ignore the chemical signals that start and stop the cell cycle. So we're going to see those cells dividing much more rapidly. So you may be wondering why we're talking about this in regards to the epithelium. The epithelium actually gives rise to the most cancers of any type of tissue in our body. And this is probably because epithelial cells divide more, so they have a higher mitosis rate, and this leads to more opportunity for damage to the growth control mechanisms. And we also see more mutations, more cancer with the epithelial cells, because epithelial cells are also, if you think of your skin's exposed to a lot of things, right? They're in contact with a lot of environmental hazards, and also ultraviolet radiation is one of those environmental hazards, other types of carcinogens. Anytime you're around harmful chemicals, things like that, touches your skin. So again, epithelial tissue is going to be the type of tissue that is going to give rise to the most types or most cancer and cancerous cells in general. Okay, going back now to those characteristics of epithelial tissue, we are on number 4. So the fourth characteristic of epithelial tissues is that epithelial tissue is what we say avascular but innervated. So what does this mean? Let's break it down. So avascular, anytime you see A in front of something, it means kind of like no, or that it doesn't have it. So avascular means no blood vessels, because the vasculature is your blood vessels, right? So avascular means that no blood cells are going to be found in epithelial tissues. So that tissue must be nourished by diffusion from underlying connective tissues. And you can see that here, right? So here's your epithelial cell, right? This is the bottom, that basal region. And if you look all through here, you're not going to see a single blood vessel. But if you look down here in your connective tissue, you see this capillary. So the epithelial tissue does not have any blood vessels, but your connective tissue does, and diffusion is going to occur up into your epithelium from that connective tissue. Epithelial tissue is going to be innervated though. And what that means, the word innervated means it's supplied by nerve fibers. So if you look here, this yellow thing, I mentioned earlier that generally bright yellow lines are going to indicate a nerve in your anatomy and physiology text. And so you can see that this nerve here passes from your connective tissue and goes up into your epithelial tissue. So it's actually in the epithelial tissue. Epithelial tissue is innervated, but it is avascular. So it has nerves, but it does not have blood vessels. Finally, the fifth big characteristic of epithelial tissues is their regeneration capacity. So epithelial tissues have a very high regeneration capacity because there might be a lot of damage. There's a lot of trauma, there's a lot of friction and other damage. And damage can be stimulated, you can see, by a lot of different things. So when that damage can cause things like losing that apical basal polarity, so losing those two sides to that membrane. Maybe the lateral contacts between the cells are broken. So maybe you see even a breakdown of those desmosomes or the tight junctions. You can often see just general friction that can lead to that damage. And then sometimes we even have exposure to hostile substances that will break down these epithelial cells. So if we have an injury or something happens to the point where that cell is even going to die. And you can see this term here, apoptosis. So apoptosis is a type of normal programmed cell death. And cell death is going to lead to basically the differentiation of viable cells. So when I say differentiation, they're starting off as just cells. They don't really know what they're doing. They have to kind of figure it out still, right? But because of that, they're able to grow, and they're able to grow to have the structure and function that is needed to be epithelial tissue. So proliferation means that they're able to regenerate in order to recover, and basically that's how your skin is able to not be open and it's able to heal really quickly. So we want to make sure that there's a very high mitotic rate so those cells can basically regrow, they can proliferate, and be able to maintain their function, which is going to be to protect, right? So your skin or epithelial tissue is going to regenerate, and they will replace with the same kind of tissue to reestablish the epithelium. You can kind of see the different steps of this regeneration process here. So you have an injury that's going to lead to that cell death, right? Then we see the de-differentiation of these viable cells. We're going to see them proliferate, so they're going to grow, right? And then they're going to recover, and they're going to reestablish that normal epithelium. So this process of regeneration is good. It's what we want. Regeneration is good. But it's also going to require adequate nutrients, and it requires the process of cell division. So I mentioned at the beginning when I introduced you to epithelial tissue that there are two different forms of it, right? So far we've talked about the form of epithelial tissue mostly that's going to cover and line, right? But in addition, remember, we have glandular epithelial. So basically a gland is one or more cells that's going to make and secrete an aqueous fluid called a secretion. And we're going to classify them by their site of product release and by the number of cells they have. So they're either unicellular or multicellular. So when we look at the site of product release, we have two different types. We have our endocrine glands. Endo meaning inner, right? So endocrine glands are going to be our ductless glands. They actually lose their ducts during the development process. So secretions are not going to be released into a duct in endocrine glands because there are no ducts, right? They're actually going to be released instead into the surrounding interstitial fluid, which is then picked up by our circulatory system. So they are going to secrete via that process of exocytosis that we talked about earlier in the semester. They're going to secrete hormones. And hormones are basically little messenger chemicals that travel through the lymph fluid or the blood to their specific target organs. So you can see over here, your endocrine glands, right? Talks about how they lose their ducts during development. They're going to secrete hormones into the interstitial fluid, and then the hormones are going to enter the blood. So you can see your endocrine gland cell here. They're going to secrete those hormones, and then it's picked up by our circulatory system. We also have exocrine glands. And exo means out, right? So these are going to be externally secreting. So think of things like sweat. So with exocrine glands, the secretions are released onto our body surfaces, such as our skin, or into body cavities. Our exocrine glands are going to be more numerous than our endocrine glands, so meaning they're much more diverse, right? And they're going to secrete products into ducts. So examples of these include things like mucus, sweat, oil. When you think oil, think of things like pimples, right? When that oil gets stuck in that secretory gland. Also, exocrine glands include things like our salivary glands. So these can be uni or multicellular. So our exocrine gland, you see right here, they're going to retain the connecting cells. So you can see this gland right here. They're going to secrete things into that duct, and then it's going to move to the epithelial surface. Again, two different types here, your endocrine and your exocrine glandular epithelia. And really, specifically what I want you to really learn about in terms of the glandular epithelium is your unicellular exocrine glands, such as your mucus cells and your goblet cells. So the exocrine glands, these mucus cells and your goblet cells are going to be unicellular. This means they have one cell, and they're found in the epithelial linings of the intestinal as well as the respiratory tract. So when you look at them underneath the microscope, or you look at them even in these pictures right here, you see these huge exocrine glands, right? And it's really just one cell. And oftentimes students get confused. They think each one of these little dots here is going to be a cell. But the reality is that it's really unicellular. There's only one cell, even though it's very large. So the specific type of secretion that we get from goblet cells and mucus cells is something called mucin. So this mucin is a sugar protein, meaning it's a glycoprotein, right? And it's going to dissolve in water, and it forms something you're probably familiar with. It forms mucus, right? Nobody likes that. Mucus is a slimy, protective, lubricating coating, and mucus occurs in our respiratory tract, right? We also are going to find it in our intestinal tracts. That's why we have something called Mucinex, right? To try to help reduce that secretion, or at least break it down a little bit. So this picture right here, what you're seeing is a goblet cell. And in a way it looks like a footprint to me, but the book talks about how it looks like a glass with a long stem, so kind of like a goblet, right? So that's how they got their name, goblet cells. So as I mentioned, it was just a short lecture today, and I want to give you some extra time to work on your epithelial poggle activity. But before I end the lecture, you can see there's a few slides left in this presentation, right? So I want to go over a few things that will help you complete the epithelial poggle activity. So feel free to pause this lecture, open up that epithelial poggle activity assignment, and you'll be able to complete some of those questions as I cover the next few slides. So I mentioned a little bit earlier that our epithelial tissue has a lot of different types, right? So classification of epithelial tissue is going to be based on both cell shape and the number of cell layers. So if we look first at the shape you see up here, so we can have squamous epithelium. So with squamous epithelium, they're very flat, right? And they're like sheet-like in appearance. We can also have cuboidal epithelium as a shape. Cuboidal cells are just like it sounds. They look like little cubes, right? So what that means is they have an equal width, height, and length, or depth to them. Then we have columnar epithelium, which again looks like columns, right? So they are taller than they are wide. So what you'll kind of notice a little bit with these different shapes, squamous, cuboidal, and columnar, notice how this dark circle in the middle of the cell, which is actually the nucleus, it conforms to the shape of the cell. So with your squamous, it's very flat. Your simple, it looks like a nice circle, kind of looks like a nucleus. Whereas with your columnar, it becomes very elongated, more oval in shape. So just keep that in mind as you're looking at different examples of epithelial tissue, that nucleus is going to generally conform to the shape of the cell. We also are going to classify our epithelial tissue based on the number of layers. So you can see the layers here. Simple, in terms of a simple layer, that means that you just have one layer. Sorry, I didn't mean, so simple, all of these up here, these are all simple. And you can see how there's just one layer, right? That's what simple means. These are typically going to be found in areas where absorption, secretion, and filtration occur. And that's why we want just one thin layer, right? So that we can have an easy route for that absorption and filtration and excretion. Then we have stratified. So you see an example of stratified here, here, and here. So in terms of stratified, it means that it is made up of more than one layer of cells. And we see stratified epithelium being very common in high abrasion areas where a lot of extra protection is needed, such as our skin. So you can see how you have multiple layers in each one of these cases for your stratified epithelium. Then we also have something that's called pseudo-stratified. And pseudo-stratified is made up of very closely packed cells that appear to be arranged in layers because they're different sizes, but it's actually just one layer of cells. That's why it's pseudo-stratified. It's a false stratification. So you can see here it looks like it's multiple layers, but really this is one cell, this is one. So that's your pseudo-stratified epithelium. So on this slide and the following slide, there are some figures from your e-text, and you should be able to review these images on the slide as well as in your text and be able to ID the structure and where they're located after you read through the e-text and after you complete that epithelial pogal tissue. So given the different shapes and types of layers of epithelial cells, there are obviously several different types of epithelial tissue, right? So I'm going to go over some of these types in this slide as well as the next one. So first we can have our simple squamous epithelium, which you see right here in A. This type of epithelium typically is going to line the blood vessels as well as the blood cavities, and it regulates the passage of substances into the underlying tissue. So it kind of resembles, if you look here, it kind of looks like a tile floor, right? And one specific place we see this is in the lungs. So that's what this photomicrograph is of. So in the lungs, it actually forms the walls of the air sacs across which gas exchange occurs. So you can see these are the air sacs of the lung tissue, and you have your squamous epithelial cells there. So again, if you kind of break down the term simple, simple means one layer, which is really useful for whenever we have absorption or secretion or filtration. Makes sense in our lungs, right? We have that carbon dioxide and oxygen exchange, right? And squamous means that they're very flat. And again, that's going to allow really good exchange across them. Then we have simple cuboidal epithelium. So this type of epithelium is generally found in glandular, meaning secreting tissue, as well as our kidney tubules. So this epithelium is actually going to form the walls of the smallest ducts of glands, and it also forms many of our kidney tubules as well. So you can see here again, simple, meaning just one layer, cuboidal. They have kind of cube-like cells, very large nucleus, good for secretion and absorption. So that's why we find them in our kidneys. Then we have our simple columnar epithelium. So this type of epithelium is often specialized for absorption, and usually it has apical cilia or microvilli. So these cells are going to line your stomach and intestines. And you can actually see here, this is this cavity inside the intestine, and you see these microvilli. And if you think about it in terms of columnar, it's a lot more surface area to it, right, compared to squamous. And that makes sense that it's in our intestine walls, right? Because in our intestines, we want to absorb a lot of things. We want to absorb those nutrients, right? And so we want that increased surface area of these taller cells. That's also why we have these microvilli, these little projections. That increases the surface area for absorption as well. So again, we're going to see these cells in stomach and our intestines. Okay, so this is kind of a continuation of the last slide. I'm going to go through 3 more pictures here. So we have our stratified squamous epithelium, which you see right here. This type of epithelium usually has protective functions, including protection against microorganisms from invading underneath the tissue, or protection against things like water loss. So the outer layer of your skin, this epidermis layer, is actually made of stratified squamous epithelial cells, and it's the most common type of stratified epithelium. You can see in this picture here, this stratified squamous epithelium. You see the basement membrane, connective tissue underneath. And this is actually from the lining of the esophagus, which is another area that we're going to see the stratified squamous epithelium. Then we have stratified cuboidal epithelium. It's actually not shown in an image here because this type of epithelium is not very common. It's going to be found in the excretory ducts of your salivary and sweat glands. We also have stratified columnar epithelium. That's also not shown in these images. So it's not very common, and it's going to be seen in your mucous membranes. So it's going to be seen in the lining of our eyelids. It's going to be both protective as well as mucus secreting. So the last picture I actually want to cover here is your pseudostratified columnar epithelium. This type of epithelium is going to line your upper respiratory tract and actually has a lot of cilia. You can see all these cilia right here. They look like little hairs, right? So the motile cilia, what they're going to do is they actually propel sheets of dust trapping mucus away from our lungs. So we want to keep them out of the lung region. Okay, so hopefully the information I just covered helps you with that epithelial poggle tissue assignment. So go ahead and finish completing that. Technically you have all week to do it, but now while it's fresh in your memory is a really good time to do that assignment. And I will see you later on for our lecture on chapter 4B. Mahalo.